talk about, perhaps in the context of kidney function in mammals. And I think most of us in this room probably associate kidney function with getting rid of waste products. That's only really part of what kidneys do. Major role that kidneys play, specifically the nephrons that we'll talk about next time in kidneys, role they play is in regulating salt and water balance in your body. Specifically, not just salt, but particular ions, regulating the balance of particular ions, in addition to getting rid of nitrogenous waste products like urea. This ability to regulate ionic balance in the body is not just the job of kidneys. You find this role performed in lots of different kinds of structures in a variety of animals. And so we'll look at how water and salt balance are maintained in marine animals that ingest seawater frequently. So here you'll see the function of salt glands, specifically things called chloride cells. We'll look at it in kidneys, but before we get to that point, we're going to start by actually looking at some terminology and defining abilities that animals have. Then we'll look at strategies, specifically animals that are referred to as osmoregulators, that is, they maintain very tight control over the total number of ions and the specific types of ions, versus those that conform. And when you're thinking about regulation and conformity, mostly you should be thinking about marine or brackish or estuarine or freshwater dwelling animals. Terrestrial animals, of course, are living in a habitat that is not wet, right? And so our osmoregulation really is tied directly to diet. We are really good osmoregulators. So when our osmotic balance changes when you eat something, we are really good at regulating that. But many of the examples I'm going to give you in terms of strategy versus conformation deal directly with animals that are immersed in a fluid that might be saltier than their body fluids in the case of salt water, or much more dilute than their body fluids in the case of animals that live in rivers and streams and lakes. Okay, so I'm going to start by just talking about the importance of body fluids generally. That's lousy. So what I'm showing you here are data, summary of data for the summary of data. Oh goodness. Of how water is distributed in a human who happens to be approximately 20 years old body, who has a body mass of about 70 kilograms. So the first thing, before going over this, is that many animals, if you ask how much of their body mass is composed of water, the answer is in the neighborhood of 50%. Lots of animals like ourselves, 60 to 65% of our body mass is composed of water. Very few animals have a lot more than that, and virtually none have less than that. So if you think about the distribution of where that water is in your body, here's a breakdown of it. So if you have a mass of 70 kilograms, about 60 to 65% of your body mass will be composed of water. And of that water, about 28 kilograms will be in intracellular fluid, that is inside your cells. I have no idea what happened to this graph. When I look at it on my computer, everything is there, but it's missing bits here. So about 28 kilograms would be in water that is inside the cells. About 12 kilograms would be in interstitial fluid. This is the fluid that kind of percolates around the outside of cells, but is not inside the circulatory system. About 4 kilograms would be in blood plasma. Plasma is the aqueous portion of your blood that does not include cells or platelets. So it's not the presence of water itself that is important. It's the presence of water and what it is doing to dilute inorganic ions and organic compounds that is important. So the role of water is really to make sure that you have not only the, let me rephrase that, you need certain ions and organic compounds in your body to be alive, to function in a physiological capacity. One of the jobs of water is to make sure that those are diluted exactly right. So if you think about sort of requirements for healthy, normal physiology. Importantly, you have to have inorganic ions and organic compounds. So examples of organic compounds are be proteins. And importantly, these have to be of the right, so the correct types, and in the correct concentrations. So the reason the correct types of ions and organic compounds and the correct concentrations are important, and we're going to focus now just specifically on the concentrations part of the statement, is that the concentrations they affect the 3D conformation of structure of proteins and DNA. For example, if you take some proteins and put them into a tube in a laboratory and you add too much salt, it'll change the structure, like they can unfold. If you dilute that sample too much, you remove too many salts, they can clump together and or unfold. The structure of a protein is critical to its function, so if you change the structure, you lose the function, or you alter the function in a way that's probably not great. Another reason why particular types of ions and their concentrations are important is that they can affect electrical gradients that are maintained across the plasma membrane. All cells maintain a difference in ions across the plasma membrane, a difference in charge, such that almost all cells have a slight negative charge compared to the outside world. That can range from -40 or -50 millivolts to -90 or even -100 millivolts, depending on the kind of cell you're talking about. If you change the types of ions that are present or their concentrations, you can change that electrical field, because many cells, not just neurons, but many cells rely on this difference in charge for communication and other cell functions. If you change that, you change the abilities of cells to do things. For example, if you increase the concentration of potassium in your bloodstream, it can lead to spontaneous changes in the abilities of muscles to contract. Right? So you can end up with seizures because all of your muscles are contracting maximally for an extended period of time. So this is really important. The third factor that's related to maintenance of correct types of ions and organic compounds and the correct concentrations deals with the fact that these can affect the osmotic pressure of cells. We're going to define osmotic pressure in a few moments, but basically it's a measure of, say, the saltiness of a solution. And if you change the osmotic pressure on a cell, you're basically changing the solute potential of the cell, which will change the water potential of the cell, which can make that cell swell or shrink, which of course can change its function. Rarely for in a positive direction. So the take-home message here is that bodies of animals, let me rephrase that animals tend to take great care to have exactly the right assemblage of ions in their body fluids at exactly the right concentrations, the right assemblage of organic compounds at exactly the right concentrations. And when those get out of balance, there are physiological mechanisms to bring them back into balance. There are some exceptions. One is, you can see in this Dungeness crab. So arthropods, like this Dungeness crab, live inside a relatively inextensible cuticle. In order to grow, they have to shed the cuticle. So what they'll do is they'll basically secrete a new cuticle inside their existing cuticle. They'll then swell with water, and they'll do that by basically absorbing salts and releasing those into their interstitial fluids and into their bloodstream. That creates a lower water potential inside. Water diffuses from their environment to the interior. That water creates a pressure as the animal is swelling, which cracks open the cuticle, and they crawl out of it. They then keep swelling, as you can see here, with water. At some point, they stop that process, they harden the cuticle, and then they basically get rid of those excess salts and return their water balance back to normal. And then they can grow into this new, larger cuticle inside. So although many animals maintain extremely tight control over these properties, there are occasions in which animals will allow those to sort of go out of balance for good reasons. Right. We'll talk about some terminology briefly. And it is related to this idea of how to describe differences in concentrations across a plasma membrane. So we'll start here with isotonic, right? These are probably familiar to you, these phrases. In an isotonic solution, you have exactly the same solute potential as you do inside the cell. So there is no difference in water potential, and there's no net movement of water across the membrane. In a hypotonic solution, you have a solute potential that is closer to zero compared to the solute potential of the cytoplasm. So there's more stuff dissolved inside the cell than there is outside the cell. That means that the water potential in here is more negative than it is out here. And so water will tend to fill the cell and it will increase in size. In a hypertonic solution, the solute potential is more negative than the cell. The cell will tend to lose water to this hypertonic solution. As we're going to use these terms a bit. When you use these terms, the perspective matters. So in this case, this is a hypotonic solution, but the cell is hypertonic relative to that solution. And so the way in which you're phrasing it is important. Another term that I want to define is osmotic pressure. As I mentioned, osmotic pressure is related to concentration differences across a permeable membrane. One way to define it is to do this experiment. So imagine you have a cylinder that is filled with fluid. And that fluid is separated by a plunger that has a little shaft on it here. And the material that this plunger is made out of is permeable to water, but not to anything else. Okay? If on the left side of the cylinder you have a salty solution, and on the right side you have pure water. The solute potential here will be more negative than over here, which means the water potential will be more negative, will be lower here than here, so water will diffuse from right to left. As it does so, it's going to increase the volume on the left side and decrease the volume on the right side, pushing the plunger from left to right. Okay, makes sense? Now imagine you take this plunger and you attach it to a force transducer that's oriented vertically, just like the one we had in lab. In this situation, if the force transducer is here, it's preventing the plunger from moving from left to right, but it is measuring the force that is applied to it as the plunger tries to move from left to right. That force is a real force, first of all. Second, that force divided by the cross-sectional area here yields you a pressure. So pressure is the force over the area, a force divided by the area over which it is applied. So in this situation, the osmotic pressure is equivalent to that force divided by the cross-sectional area over which that force is applied. Osmotic pressures are real. You can actually measure them as pressures inside the bodies of animals, okay, mostly across the plasma membranes of cells, but they are real, which is why we're using them. But just for shorthand, they are not equal to, they are directly proportional to concentration differences across membranes. Okay, so I want to talk now about different strategies that animals use with respect to regulating or not concentrations in ions, organic molecules, and water. So the three types we're going to look at. The first is called osmotic regulation or osmotic conformity. And then we'll come back and look at ionic regulation and volume regulation. All right, so the context for this is, imagine you have an animal that is aquatic. It's living submerged. It's not you. We are not good candidates for this kind of experiment because our skin is basically waterproof. Not completely, which you know if you spend too long in the tub or swimming pool, right, your fingers shrivel, which represents a change in water balance, but more or less we're impermeable. So let's think now about fish, which have gills that are highly permeable and can lose or gain salts or water, or an invertebrate that can lose or gain salts and water across its body wall. And let's take that animal and put it into a container in which we can adjust the salinity of that container. And we're going to report those units of salinity in units of osmotic pressure. And these units are in millismoles, which are the really strange units that one uses for osmotic pressure. 0 represents pure water, and 1,000 represents the osmotic pressure of seawater. So anything in between, fresh water in a stream is going to be in the single digits, maybe 6 to 10 milliosmoles. Everything else in between is going to be brackish water that you would find in an estuary. Okay? Let's look at the vertical axis. This is the osmotic pressure of the blood. How concentrated is the blood? All right, let's start on the right here. Some animals are osmoconformers. That is, if the environment they're living in is at, say 400 milliosmoles osmotic pressure, their body fluids will match that exactly. Have exactly the same osmotic pressure. This dashed line represents the line with the slope of 1. Okay, so osmoconformers conform to the environment. Osmoregulators are illustrated here. Here you see an animal who is maintaining its blood osmotic pressure at a given level, regardless of the change in salinity in the solution it's living in. Let me show you some examples of real animals, not those two extremes. So some animals, like this blue mussel, are excellent osmoconformers. It doesn't matter what the salinity is in the water they're living in, their body fluids match it exactly. In other words, their body fluids will have the same solute potential as the seawater they're living in. Other animals, like this shrimp, are pretty good osmoregulators. Their body fluids increase very slightly in osmotic pressure as you increase the salinity in the water they're living in. And then only once you get above full strength seawater does their body fluids start to get a little bit salty. Lots of animals, however, are like this green crab, which is they're very good osmoregulators at some limited set of environmental salinities. As you increase the salinity around them, they're still regulating, but they're starting to conform a little bit. And then at high salinities, they become very good osmoconformers. Right, so that's actually not uncommon to see in some animals. Next, I'd like to look at ionic regulation or conformity. The difference between osmotic regulation and ionic regulation is, in osmotic regulation, you are maintaining, if you're a regulator, a different overall concentration of salts and other things in your body, regardless of what is happening in the environment. In ionic regulation, you are regulating the concentrations of very specific ions, and you may or may not be an osmoconformer or regulator. You can see that on this table. So let's focus your attention just at the top first. So what you're seeing here in the top line, we're going to measure osmotic pressure in units of milliosmoles. And over here you see a partial list of ions that are really important in physiology and also are found in seawater. Right, so let's look at seawater first. The osmotic pressure of seawater is 1,000 milliosmoles approximately. Okay, that's full strength seawater. And if you look at the breakdown, the ionic breakdown, you'll see that seawater has 460 millimoles per liter of sodium, 10 of potassium, 10 of calcium, 53 of magnesium, 540 for chloride, has some sulfates in it, not a lot of phosphates, and no urea. Okay. Let's look first at a hagfish now. A hagfish is, it's not a bony fish, it's a jawless fish. They look kind of like big eels, they're really disgusting, they produce a lot of slime. They're found mostly in deep sea, although they come up to relatively shallow waters at night. If you measure the osmotic pressure of the blood of a hagfish, you'll see it's almost identical to that of seawater. This is an osmoconformer. And if you look at the ionic composition of its blood, you'll see that it's approximately an ionic regulator, but not a very good one. Right? So a little bit more sodium than seawater, a little bit less potassium, a little bit less calcium, magnesium, and so forth. Okay? They have a little bit of urea in their blood. They have some phosphates and fewer sulfates than seawater. Let's look at a shark. A shark is also a very good osmotic conformer. Its body fluids are close to that of seawater. Not exactly, but very close. But they are really strong ionic regulators. So they have 2/3 as much sodium as you find in seawater. They're clearly lowering their blood sodium relative to the seawater they're living in. And you can see much different concentrations of these other ions. And they have a heck of a lot of urea. This tells you how they're becoming how they're becoming osmotic conformers. They generally have lower concentrations of these ions than seawater. The way they're increasing their osmotic pressure is by having urea in the blood. Urea is a metabolic waste product from the metabolism of amino acids and nucleic acids. How are they keeping this high? Well, they drink seawater every time they swallow prey. Right? So their body fluids are influenced by the seawater they ingest, as is the case for hagfish. Every time they eat something, they ingest seawater. And that is how they're exposed to the ions that are present. Let's close by looking at this really strong osmoregulator, which is a bony fish, it's a flounder. What you see is that flounders maintain a blood osmotic pressure that is a third that of normal seawater. So these are not osmoconformers, they're osmoregulators. And you can see that they also are very strongly regulating various ions. So a flounder is an example of an animal that is an osmoregulator and an ionic regulator. A shark is an example of an animal that is an osmoconformer and an ionic regulator. Whereas a hagfish is an osmoconformer and more or less an an ionic conformer. The very last thing we'll talk about is volume regulation or conformity. This one's pretty obvious. All this refers to is the total volume of water in the body. So, imagine you drink 2 gallons of water. Where is most of that water going to end up? So let's back up a second. Here. There, see, it's not a screwed up slide. It has all the parts. Go ahead and fill in. So if we look at this, and let's say you drink 2 gallons of water, where is that water going to end up? First of all, what's going to happen to you if you drink 2 gallons of water? Well, where will you spend a lot of your day?
The hospital.
That's right. Excess urine production. So where does most of that water go when you're drinking it? Not in here. Actually, right here. Because we are really good at volume regulation. So you drink the water crosses your intestinal wall into the bloodstream. Your body measures the increase in volume, the decrease in ionic concentration, and the decrease in osmotic pressure, and your kidneys spring into action and get rid of that water. Right? So that's an example of, the fact that animals also are measuring volume. So these three things are different. They're distinct from each other, right? Osmoregulation is distinct from ionic regulation, which is distinct from volume regulation. But they are almost always intertwined. If this freshwater fish, eats something, it's going to drink some fresh water. Its cells and interstitial fluids are more concentrated than the water it's living in. So in the example I just gave with us, it's the same thing with this fish. It drinks some water. The first thing that does, right, is it increases the volume of the blood plasma. Because you now have more water but the same amount of salts, you've diluted the ions that are present. And because you've diluted the ions, the overall osmotic pressure decreases. Right, so you almost can never change one of these without changing the others. Rephrase that. You can't change the volume of blood through ingestion of water or taking water across the body wall without also changing the others. These are all interrelated. All right. So I'm going to close with some tragic news. We didn't get to salt glands, so they're not in the exam. It's a bummer. Such good questions I had for salt glands. It was going to be half the exam, maybe more. Now it won't be present. So I will send out an updated study guide, which will remove the bits about salt glands and chloride cells. We are not meeting on Tuesday for lecture. Look to Canvas for a narrated PowerPoint. And if you have questions, I'm sure come